Hi, I'm Raghavendra Rao. I'm here to talk about on radioactive carbon dating. So you are able to see a guy whose name is Willard Libby. His innovations in 1946 regarding the finding or the determining the age of the object containing organic material by using properties of radiocarbon or isotopes of carbon. So he used carbon 14 as the one of the carbon to find out the age of the object. Basically, his contributions are in general were recognized and he has been awarded Nobel Prize in 1960. When we talk about this carbon, where this carbon is in general integrated in your body. So we all know that the primary staple food for all living organism is in general carbohydrate. Basically, eventually when we eat carbohydrate substances, the carbon which is present will be retained in the macromolecules. We have macromolecules, so we have macromolecules like nucleic acids, proteins, carbohydrates. All these are in general considered lipids are considered to be the macromolecules. So in nature, there are three types of carbon are present. One is C12 and another one is C13 and third one is in general we consider it as C14. Basically, each of these are available in the nature, but when we look into the molecular structures of these three, they differ in their electrons are similar in all these things, but the protons and the neutrons are differ. So basically, we consider the isotopes of the carbon. So majoritarily we will see carbon 12 in the nature, which is consists of 6 protons and 6 neutrons, and it is considered to be the stable, and its percentage in the nature is 99% of the carbon made up of C12. So next to that we have C13, which also consists of 6 electrons, 6 protons as well as 7 neutrons, and it's also a stable, and its percentage in the nature is 1.11. Basically, the stable nature of these two is nothing but when they are integrated into the molecule, once they form, are utilized for the formation of structures, so they cannot be separated from that. They will be the integral part, and you cannot be separate those carbons which are making the macromolecules and other molecules. But when we look into the eventually the third carbon, which is C14, basically it consists of 6 protons and 8 neutrons, and it is considered to be one of the isotope of carbon. It is in general observed in the nature by the reactions. So when the cosmic rays basically react with the nitrogen atom, so these nitrogen atoms are get converted into C14. Basically, it is not only that the radioactive substances are cosmogenic substances and anthropogenic activities. All these are in general may help in the conversion of nitrogen into C14. So this is a reaction which I have shown here, how the cosmic rays reaction will convert the N14 to C14. Basically, when the nitrogen atoms absorb the cosmic rays, they lose the neutrons. Due to that, what happens? It converts into C14. Vice versa, when the C14 molecule liberate the beta particle, they eventually get converted back to the N14. So this is what happens, the conversion of nitrogen into C14, and again the C14 will get back to N14 by the liberation of beta cells. So in this way, you will observe the stability and instability of carbon and related isotopes. Now, when we look into this, basically you can see, as I mentioned, the majority of the portion in the atmosphere is C12. Basically, the carbon dioxide molecule consists of two carbon, means it consists of two oxygen molecules. Sorry, it is not carbon. two oxygen molecule and the central carbon, and 99% of the carbon dioxide containing carbon is of C12. So they will be utilized by the plant for the process of photosynthesis. Accidentally, along with the C12 and C13, you are able to also see in the nature the C14 carbon. So here what happens, this C14 is also absorbed by the plants for the purpose of making food material in the form of photosynthesis. Both are eventually will be forming in the food. When this food is eaten by the individual, basically you will see both the carbon atoms are present in the human, where they are utilized for the making of protoplasm from their various macromolecules are in general formed by utilizing the stable C14, means unstable C14, stable C12 and C13 carbons. So here has been represented only two types of carbon. The red color represents the stable one and the green color represents the C14. So now here the amount of C14 which is present in the atmosphere is equal to the amount of C14 which is present in the human individual, that is the body. So because he consistently takes up the C14 which is available in the atmosphere for the purpose of making macromolecules by eating the food material, so he always maintains the ratio of C14 in the atmosphere with the C14 present in the body. Similarly, the ratio between the C12 in the atmosphere and the C12 in the body is also will be maintained. If suppose eventually due to some calamity or disease the person died, so it has took place. Now what happens? The person will stop eating the food. Because of that, there won't be further increasing in the C14. So in this way, the C14 will be stopped by the individual due to his or her death. Now what happens? As I have mentioned, the C12 is a stable one which cannot be removed from the macromolecule, but the C14 is unstable. It gradually start decaying, means the ratio between the C14 in the atmosphere inside the dead body will be display, means there is a difference in the ratio of these molecules of carbon when we look into the atmosphere as well as in the dead. Now when we look into the dead body, so what makes the decay of C14 molecules in your dead body? So basically we all know that the C14 molecules liberate beta particles. Now you are able to see when you keep a dead body in a chamber, what you are able to see, this is a chamber, and this chamber, when you keep the dead body in the chamber, now you are able to see these are all the beta particles which are emitted from the radioactive C14. So these emitted beta particles which are released from the dead body is decaying the C14 content in the dead body. So when we look into the half-life period of the C14, at 0 time the individual will have 100% C14. So there won't be after any intake of C14 by the dead individual. So that's why you will always see there is a decrease in the concentration of C14. So by the increasing of the years. So there will be 50% decrease when the dead body age becomes 5730 years. So like this, here 11,467 is the year. So in this way, what happens here, basically you can see that the half-life period can be 5730. Gradually the decay of this C14 in the body of the individual after the death will be like this. So these are the different stages. At what time, what will be the percentage of C4 which is present in the dead body. So basically when the dead body becomes fossil, when you find this dead body in the excavations, when you collect a piece of sample from it and you test for the presence of C14, so basically you will end up with a finding out of the age of this particular object, which is in general having the organic substances in its body. So based on that, we calculate the or estimate the determine the age of that particular object which contains organic substances, that is of living matter. Basically when we look into this, at time 0, you will see there is 100% of C14 will be there. So gradually, when it decreases to 5760 years later, when we observe it is of 50%. Then if it is, when we observe after the death of 11,460 years, you are able to see 25% of the C14 carbon. And after the death of 22,920 years, you would be eventually finding out 12.5% of the C14. And if you just find 20 2,920 years later of the death, you will be finding out 6.25% of the C14. Like this, if you find the dead body after 28 years, 28,650, you are able to see nearly 3.125 percentage of C14. So you don't get never to zero here. It is always half the way to reach the wall, like that. It never come down to zero in this way. So in this way you can finding out, suppose you have recently found a dead body. You have seen a dead body recently which has died of a disease. When we find out the presence of C14 in this, and if it contains nearly about 37% of the C14 is present, that eventually what happens, it comes nearly about this place. And normally you can estimate the age in between the 25 to 50. That's why you can calculate based on the amount of C14 and the year. We can, based on the ratio, you can estimate or determine the age of that particular dead body or the organic body or substance which is present in this. In this way you can see, so there is a decrease of this linear regression in this, where you can see this is a half the way you can see at the times versus the percentage of C14 present in the dead body or objects. Now, when we look into that, we all know that the C14, by liberating a beta particle, it gets converted again back to nitrogen. So, in this way, you can see after the 5700 years, eventually you will see the NC14 will get converted into N14 50%. Like this, the exponential growth can be seen in the increasing of N14 in the atmosphere, because it get converts C14 to N14 by liberation of beta particles from the object. So in this way, So we can find out the formation of C14 from the N14 and reversible reaction of C14 to N14 in the nature. So now when we look into the accuracy of this, so basically what we can say that when we take a known object and its age, and if you observe the C14 of such material, eventually we can find out how much old is this object. We already know. If the C14 carbon dating technique, if end up with the same results, then we can authentically say that the accuracy is there in the carbon dating technique that is based on C14 carbon dating techniques. So this is the way basically we see the accuracy of this particular carbon dating technique. So now coming to the this graph, which is you are able to see the wandering effect, where this blue line indicates the natural levels of C14 in the atmosphere. So after 1950s, you are able to see there are two large peaks which have been developed in the southern hemisphere as well as northern hemisphere. So you can see in the southern hemisphere and the northern hemisphere, like what we can say in the southern hemisphere, in the Wellington, that you in the New Zealand, which has been raised about 160 parts per million, and similarly in northern hemisphere you would reach to the 200 ppm in the Austria. This massive increase in between 1953 to 19 what we can say 65. This is mainly because which we call it as a bombing effect. We call it as a bombing effect. In this, most of the countries they started using nuclear weapons test. So this nuclear weapon test has eventually increased the concentration of radioactive carbon. So due to this, basically the accuracy can be tested to those objects which are fossilized before 1950. And if you just test for the determination of age after the 1950, basically due to the high nature of the radioactive carbon in the atmosphere, you may not end up with the accurate age. And also, so this carbon dating technique cannot be used for those fossils which are of very old in nature, beyond 60,000 years. So when we go for some millions of years and some above the 60,000 years of objects, basically you may not get accurate results, and this carbon dating technology will not suit to this. We need to use some other alternative dating techniques which normally gives you the age of these things accurately. So this is how normally the carbon dating technology is used to determine the age of the objects which contain organic substances.